The difference between biological and technical replicates. Technical replicates are repeated samples taken from the same source. For example, 3 blood samples from the same individual to check whether your experimental technique is reproducible. Whereas a biological replicate refers to sampling from different sources, like for instance taking a blood sample from 3 different individuals to see whether the result overall is biologically reproducible. Okay, I have cells that I'm going to treat with chemical X, and I want to compare the growth rate to just normal, untreated cells. Oh wait, this is only one dish, so we're gonna need at least 3 of each, right? So can I just use 3 different wells of this 6-well plate here for each condition? Or maybe 3 different plates of 3 wells each? That seems like a lot. Well, wait a second. The definition is that each individual is a biological replicate. So like, does each cell count as a biological replicate? Hey, welcome to Science is You. Whether you're a student in grad school or just want to learn more about science, knowing how to check whether data is repeatable or not is critical for setting up your own experiments and for evaluating the quality of data you come across elsewhere. Biological and technical replicates cause a lot of confusion. These terms can be neatly defined in theoretical examples, but in reality things can quickly become frustrating for a number of reasons. First, the same logic might not apply one-to-one for different types of experiments, like blood samples and cells in a dish. Also, we have to consider the doability of experiments. In theory, it might be great to repeat an experiment 1,000 times to make sure the data are consistent, but you'd be in the lab for 10 years. Another consideration is how important the experiment is, meaning how critical the observation is for the point you are making, and or how much the topic has been previously studied. For example, if you believe that a brand new drug formulation reduces tumor size by half in mice, it's probably a good idea to devote more time on this experiment than to verifying that the mouse tumors are also reduced by traditional radiation treatments. The latter could be relevant, sure, but it just isn't the main point of the paper. Finally, for all these reasons, you will run into some differences of opinion regarding what constitutes a biological or a technical replicate and how many replicates need to be analyzed. But let's think about it differently. A replicate simply means repeating something to see if you get a similar outcome. Now, how many times we need to repeat the experiment is a topic for a later video. But generally, an experiment needs to be performed at least 3 times to generate 3 independent biological replicates. But what exactly constitutes an independent biological replicate in the context of cell culture? Is it 3 different plates of cells, 3 different wells of cells on the same plate, 3 samplings from different areas of the same dish? Can you set up the 3 plates all on the same day, from the same stock of cells, or should it be on different days from different stocks of cells, different cell types? Instead of thinking about everything in terms of a type of replicate, let's think about the different major steps of the experiment where we might have sources of error, and what's doable to check for. Imagine you're baking cookies. Okay, bear with me. For the sake of argument, let's say you're preparing for a very competitive bake-off or something. Your goal is to see if adding peppermint extract improves your cookie recipe. You purchase the ingredients, mix up the batter, and separate it in half. To one half, you add the peppermint. Then you portion both batters off onto baking sheets and cook them. If we made an error somewhere in this entire process, what errors are most likely to ruin the whole taste test? There could be something wrong with our ingredients. For example, maybe the milk we used was spoiled. Then there's making the batter. Say we mixed up the salt and sugar. Now both types of cookies taste about the same. Terrible. Say perhaps we made a less devastating mistake and added 1 tablespoon peppermint extract instead of 1 teaspoon when mixing the batters. Another error might be we didn't portion the cookies equally on the baking sheet, so a few were smaller and burned. Finally, maybe there's a clump of dry flour in one of the cookies. So there are 4 main places here where we might have errors: purchasing the ingredients, making the batters, portioning and baking each cookie, and imperfections within the individual cookies. Some of these errors are easier to make and to go unnoticed than the others, and some errors will impact the final result more than others. For example, a clump of dry flour in a bite-sized cookie might not be as detrimental as mixing up the salt and sugar in the whole batch. Perhaps then, breaking each cookie into pieces and tasting each piece is a bit unnecessary. Alternatively, something wrong with our ingredients like spoiled milk would really skew our results. But it's pretty easy to identify if something like that's amiss, and there are checks we can do beforehand, like the expiration date, sniff test, etc. It probably isn't worth the cost and waste of purchasing 3 different sets of ingredients from 3 different stores just to prove that point. But errors in mixing the batter, teaspoons versus tablespoons for example, might make the peppermint cookies taste weird, whereas in reality we just accidentally put in way too much and didn't notice it. Mixing the batters correctly is critical to being able to get a result from this experiment, and errors here could easily go unnoticed. If we repeat the batch 3 times, then it's unlikely that we'd make the same mistake each time. So mixing the batter is our biological replicate. Wait, what about inconsistencies between the individual cookies? Well, if you get a burned cookie from the peppermint batch and a well-cooked one from the plain, then that could definitely skew our results in that one batch. What we could do then to ensure we're getting a representative sampling from the cookies is taste 3 of each type per batch. These technical replicates check for error within batches or within the biological replicates. Believe it or not, experiments with cells are the same thing. You have your initial ingredients and stocks of cells, then you mix your solutions of cells with nutrients and the chemicals you're testing. Then you plate multiple wells of each. This whole process would be one batch. On a different day, you repeat the procedure for a second biological replicate and so on. Why a different day? Well, thinking back to the cookies, if we line up 3 different bowls, then it becomes way easier to make the same mistake for all 3 batches because we're doing it all at once. We'd just go down the line using our tablespoon instead of our teaspoon or whatever. Different days for cell culture is because usually it takes cells at least a day to grow after being put on the plates. Theoretically, different biological replicates could be done on the same day, but if any steps for the replicates are done in parallel, it makes it easier to propagate errors and it wouldn't count as a biological replicate. Well, what about differences within the cells in each plate? Well, remember the bite-sized cookie. Even if there is a small patch of unhealthy cells right there, if you are collecting and analyzing the DNA or RNA from all the cells in the entire well, then it probably won't contribute much to the outcome. However, if you're looking at a piece of brain tissue or something where you aren't sure whether it has the same structure and composition throughout, it makes sense to look at differences within the same sample. Let's try another example. Say you're trying to compare 2 tests for detecting cholesterol in blood and see which one gives more consistent results. In this case, your question is about the consistency of the test, not anything necessarily about the specific individual. Here, it would make sense to measure 3 different samples taken from the same individual, because if you just take one sample from 3 different individuals, you don't really know if any variation is due to the test not being consistent or differences between individuals. But let's say you change your question and want to ask whether people who eat 1 piece of miracle fruit every day have lower cholesterol than people who don't. In this case, you're interested in what's going on in the people, not the test itself. Taking 3 samples from the same person still gives you just one person as a data point, fruit eater or non-fruit eater. And one might just happen to have lower cholesterol than the other. Here we'd have to look at groups of lots of different individuals to see if eating the fruit really correlates with lower cholesterol across multiple people. The key takeaway here is not to get caught up in the terminology and definitions of biological versus technical replicates. Most important is to critically think through the question you're asking and where in your experiment errors or variation could change your answer the most. This is the part you should repeat. And let's end on a question. How many times do you think an experiment or key step should be repeated? Please share in the comments. Don't forget to subscribe to help this channel get off the ground. See you next time, and remember,